It's my pleasure to start the room 4 of the session 2 of today's meeting. And my name is Anton Naumov, and I will be a session chair today. And it is my true pleasure to introduce Sarah Bond with her talk on experience with extrinsic rewards that does not undermine intrinsic motivation in rats. Thank you.
All right, so I'll be talking about motivation in rats. And to get into some of the background, I did marching band for 8 years, and prior to receiving any award or reward for practicing, I just intrinsically enjoyed practicing and just improving and just for doing it for just practicing. And once I got to college, I started to get grades for how I played and how well I was doing as far as practicing goes. And so once I had started to receive, an extrinsic reward or a grade, and going back to just not receiving one after, having received that, I wasn't as intrinsically motivated to just practice because I loved practicing. And so that is basically what the overjustification hypothesis is, where following an extrinsic reward, that behavior or the intrinsic motivation to engage in that behavior is reduced. And so Leper, Green, and Nisbet found this in preschool children where the intrinsic behavior was drawing. And so a baseline period of how long they drew was measured. A manipulation where they either expected the reward, did not expect it, or did not have a reward was in place. And then a baseline period where, again, they were just there, how much they drew, free drew was measured. And so children that expected the reward drew, spent less time drawing than those that were in the other conditions. And so the overjustification effect was found in this study. And so the effect overall is not very simple. When looking at this graph here, or this diagram here, it's just the overjustification effect, or not the overjustification effect, but intrinsic motivation. And so when looking here, the plus means that intrinsic motivation increased. And so that means that the overjustification effect did not occur. And so in low interest tasks, following a reward, intrinsic motivation increases, so it doesn't occur again, the overjustification effect. However, with high interest tasks, such as verbal behaviors, such as saying good job, that being the reinforcement, the intrinsic motivation increases. So again, the overjustification effect is not found. However, for just doing tasks for such as the children's study where they were drawing and just doing the task resulted in reinforcement, intrinsic motivation decreases, so the overjustification effect is found. And so there are cognitive and behavioral explanations for this, and the cognitive aspect of this is that cognitive dissonance could occur where you think that you're intrinsically motivated to do things that you like or just in general, but you realize that following a reward or just, having experienced an extrinsic reward results in, maybe you're not as intrinsically motivated as you thought. And so we will be focusing on the behavioral aspect using lever pressing in rats, and using rats will eliminate any explanations that are cognitive. And so in previous research with rat lever pressing, it has been found that pressing a lever occurs at a low rate in the absence of an extrinsic reinforcer or an extrinsic reward. And so therefore that can be used as a measure of an intrinsic behavior. And so other research has found that following lever pressing, if the light is eliminated, that also maintains a level of lever pressing, as the light and that change in the stimuli results in, just motivation to continue to do that behavior, such that if the children didn't have any ink coming out of their markers when they were drawing, it wouldn't be very motivating because the stimuli around them aren't changing due to their behavior. And so lever pressing was our measure of intrinsic motivation. There were 16 Long Evans hooded rats. There was an extrinsic group where they received food in the manipulation period, a control group where they didn't receive food, and a baseline of how much they responded prior to that, and a baseline of how much they were lever pressing following that. And we would predict that those that experienced the extrinsic reward would respond less at those than those that did not, as that would indicate the overjustification effect. And so we did not find that The extrinsic reward group did respond significantly more or press significantly more than the control group, and we would have expected them to press significantly less than the control group if the overjustification effect would have occurred. And that there was a significant effective group in just the phase 3, just the second baseline measure, and an effective day. And this is collapsing into blocks of 2 session blocks, and that effect is present there as well. But we wanted to make sure that the rats in the extrinsic group weren't just high pressers and that there weren't individual differences. So we switched the groups where the control group now received the extrinsic reward, and the extrinsic group then received the control group. And we found the same thing where the control group, what was the control group was responding more than the extrinsic group, but this was not significant, but visually the effect maintained. And so what does this mean? We did not find the overjustification effect, but previous experience with the extrinsic reward, or the food in this case, did enhance responding to the lever. And so some things that this could mean, or what this could mean, is that maybe lever pressing is just a low interest task, or it's not very intrinsically motivating to a rat. And that could be why, following that extrinsic reward, the intrinsic motivation was increased for pressing. And so
thank you, Sarah. We're gonna have to wrap that up. Were there any questions and anybody had to send in the, you can send them in the chat to one of the hosts, or you can ask out loud if there are any questions.
yes, Anna.
Go ahead, Anna.
Oh, okay, thank you. Yeah, I just wanted to ask, so if you wanted to find a higher interest task for a rat, what would you choose?
That's a great question. And I'm honestly not sure how to answer it, because when I think of behavior in rats, where lever pressing is just kind of what I'm familiar with. And so I'm not sure, but that is a good question. It's something good to think about when continuing this line of research.
Thank you for your question. Okay. Thank you very much, Sarah.
Thank you.
How's it going?
Anton, you want to tell us about that?
Hey, can you hear me?
Yeah, now Josh, are you with us? Excellent. You should be co-host. You can share your screen.
Okay.
I understand, Anton.
Sorry about that. I came straight from another presentation. So screen number, let's do 2. Share.
Right. Yeah, Derek, I think now when you're the host, you have to unmute me.
Oh.
Because I cannot unmute myself. That's the issue.
Okay. Can you hear me?
Yes, yes, yes.
Josh,
Okay.
thank you.
George, thank
Okay.
you so much. So I would like to introduce George Davidson with the presentation of identifying outcrops of the Eagleford Shale and Woodbine Sandstone in the Dallas-Fort Worth Metroplex using GIS.
Oh, thank you. So first of all, everyone can see my poster, right? Okay.
Yes, we can.
Okay, thank you. So I'm going to go ahead and get started. So the study is actually, it's called Evaluating Eagleford Outcrops Within the Dallas-Fort Worth Metroplex. And I actually am just focusing on Eagleford. So it must have changed since when I posted my abstract the first time and now. But so I guess I'll jump right in. So there are a lot of major geologic outcrops within the DFW Metroplex. And the major formation that I'm going to be talking about is the Eagleford, which is a clay-rich mud rock that can actually pose geological hazards due to the nature of its shrinking and swelling, and can cause foundation issues for homes and commercial development and roads and other things. So in the study, I I produced a geologic hazard map that illustrates all areas where the Eagleford reaches the surface in DFW. So a little geologic background. So the Eagleford Shale was deposited in the late Cretaceous around 100 million years ago. And this deposition coincided with a max transgression, which flooded the mid-continent of present-day US and basically established the Western Interior Seaway. This Eagleford Shale, it contains bentonite, which is a clay made from volcanic ash. And within this clay is a mineral called smectite, which is subject to swelling and contraction in tandem with absorbing and losing water, which can cause major foundation issues. So the objective here is to find all areas where Eagleford reaches the surface and display these areas on a map. And the map will illustrate zones that may be hazardous for commercial development or infrastructure and what have you. So some of the data that I use, so I use the Geologic Database of Texas, which was created by the BEG, Bureau of Economic Geology, and the United States Geological Survey, and the Texas Water Development Board. And together, these three entities developed a statewide compilation of surficial geologic data containing 117,000 rock units, 16,000 member formations, and 11,000 faults, and 554 other unique geologic symbols. This geodatabase is made up of a number of featured data sets that correspond to 4 different resolutions. The one that I used in this study was Geology 250K, which is this right here in this red square. And this is a map of the Geologic Database of Texas. So some of my methods. So I first uploaded the geodatabase Texas map to ArcMap. And I then had to match the symbol style to that of the Geologic Database of Texas, because the symbols they give you in ArcMap are the default symbols, and they don't match. So then I uploaded a raster polygon, or I'm sorry, not a raster polygon, a raster file to ArcMap of the counties within the north central Texas, and I clipped the geodatabase to the study area, which is what you see here. I then added road networks, Texas road networks, to the study area, and I created a buffer around them and intersected all roads with Eagleford outcrop to kind of show where the roads are and where they intersect with the Eagleford. I then geotagged photos of known outcrops to a web map, basically to substantiate the validity of the geologic map. And that's what you see down here. You see basically a geotagged image to Eagleford outcrop in the Bachman Creek area in North Dallas. This photo was taken by Dr. Richard Din, and I geotagged some other ones, but this was probably the best one that I could show for. So as a result, I developed this hazard zonation map that you see here. And the Eagleford is located within this kind of green strand right here in the middle. And the symbol, I don't know if you can see it, but it's green and has these like white diagonal lines that go through it. And these dark blue lines are all the road networks that intersect with Eagleford outcrop. So within this boundary, there are 667 miles of road networks and 1,305 square kilometers, and the population within these areas, according to the United States Census Bureau, was 847,000 people. And this kind of goes outside the DFW Metroplex. It's really more north central Texas. And there's also 336,000 single family homes within this boundary. So I guess, what do you do? What are some solutions? Because this is also obviously a massive area, and you know, it's also right in the middle, right in the heart of DFW. So I guess if you are a homeowner, main thing you want to do is maintain moisture levels, maybe water your maybe run your sprinkler systems twice a day in a time of drought. Maybe plant your trees away from the house so they're not competing with the soil around the foundation of your home. Drain water away from the house so it's not collecting around the foundation of your home, and embed the foundation so you just keep a proper amount of compacted soil around the foundation to help soil retain its moisture during a dry month. So to conclude this, Eagleford outcrop is very present in this part of Texas. And it's definitely something you want to keep in mind. Government entities or construction development, or before you commence in development, you want to keep that in mind. So, and that's it for me, guys. Thank you.
Thank you so much. We have just a little bit time for questions.
Okay.
Anyone who has a question? Yes, Amy.
I think that was just clapping.
Ah, okay. Any questions? Okay, then I've got a question. So you were talking about the volcanic ash. So it's interesting from the standpoint of a totally non-professional in this area. When were the volcanoes around here? How long ago did we have the volcanoes around here?
So I am not entirely sure when there were volcanoes around here. Trying to think of a time of volcanic activity. Probably, I would say, and Dr. Busby can correct me, I would say the Jurassic. So, I mean, and when this was deposited, it was in the Cretaceous. So I assume around that time, that's where the volcanic ash would have been coming from.
Very interesting. Thank you so much.
Thank you.
Thank you, Josh. And our next speaker, Brooke Newell, should be ready to load.
Okay. Is that all? Thank you, guys.
Thank you so much.
Have a good one.
Thanks, Josh.
Thanks, guys.
Hello. I'll just share my screen.
That looks good. We can see it.
All right.
Wonderful. So I am happy to introduce Brooke Newell with her talk on side effects of sorption of nanomaterials on iron oxides. Go ahead.
Awesome. Well, some of you guys might be wondering, what exactly are nanomaterials? Well, if you have recently received the vaccine, congratulations, you have been a part of nanomaterial technology. mRNA COVID vaccines use nanomaterials in their vaccine delivery. But beyond just vaccines, nanomaterials are becoming increasingly common. And with their increased use, more of them are entering the environment, which makes it important for us to be able to understand how they're interacting with minerals in our environment. I'm specifically looking at the nanomaterial PAMAM and how it's interacting with the common iron oxide ferrihydrite. I want to know how size is affecting these PAMAM interactions, as they come in various different sizes for generations. Basically, the higher the generation, the larger the PAMAM. I'm specifically looking at the smaller generation 1.5 PAMAM and comparing it to the larger generation 3.5 PAMAM. As you can see, they are carboxylic acid terminated. And in order to make sure that size was the only difference between my PAMAMs, I made sure to hold the number of carboxylic acid groups constant, as well as run all of my experiments at a pH 5. Here you can see my experimental setup, where I basically ran a PAMAM solution through a tube containing my ferrihydrite and measured the effluent absorbance. This allowed me to determine how much PAMAM was going onto the ferrihydrite, how fast it was going on, and how the reaction was progressing. Quantity, rate, and dynamics. Let's look at the sorption and the desorption. Here on the sorption graph, you can see that as the curve is rising, PAMAM is going onto the ferrihydrite, and as it levels off here at the top, all of the PAMAM that's going to go onto the ferrihydrite has gone on. Here on the desorption phase, as the curve is going downwards, PAMAM is coming off, but as it flattens off, no more PAMAM is coming off of the ferrihydrite. I ran this at 3 different wavelengths, and all 3 of them showed a very similar trend, which lets me know that any of these wavelengths are good to be able to measure this reaction by. And so for further research, I can use any of these wavelengths. Now, the quantity sorbed ended up being the same for both the 3.5 and the 1.5, which lets me know that size is not affecting how much is going onto the ferrihydrite. This is probably more likely due to the carboxylic acid groups, and since I held that constant, it makes sense that there is no sort of difference for how much is going on. What was different, though, was the dynamics of the reaction. So I found that it was going on in 2 separate phases, a first faster surface reaction and a second slower diffusion reaction, where it was actually going into pores within the ferrihydrite. If you look here at the fitted model parameters, you see that for the 1.5, about half of it was that surface reaction, and about half of it was that diffusion reaction. This is compared to the 3.5, where 75% was the surface reaction and 25% was the diffusion reaction. You can tell that because it's bigger, it's not fitting into those pores as well, so therefore not nearly as much of it is going on during that slower diffusion reaction. If we look over here, we can see the rate of the reaction. For 3.5, it has this sharp curve, which lets me know that most of it is going on in that faster surface reaction, compared to the 1.5, which has a much softer curve and tails off at the end. It's a longer reaction, so therefore more of it is happening in that slower diffusion reaction. As far as desorption, here we see that the 3.5, for 83% of it was coming off in that desorption phase, compared to only 48% for the 1.5. So we know that half of it is still stuck within those pores of the ferrihydrite. So how do I know the reaction is, and the desorption reaction is only on the surface? Well, that's because of the dynamics. So if we look here, there's only one part of the reaction, that faster surface reaction. If we look at the rates, we see that the 1.5 is actually coming off faster than the 3.5. Although the same amount was originally put onto the ferrihydrite, it makes sense that the 1.5 is coming off faster because the proportion on the surface is less. So since there's less of it on the surface compared to the 3.5, it's going to come off faster. So size is obviously making a difference here. Let's look exactly what's happening on the ferrihydrite. For the 1.5, because of its smaller size, when it goes onto the ferrihydrite, it's going both onto the surface and into these pores. And when we go to take it off, what's on the surface is coming off, but we're leaving behind what's trapped in those pores. As far as the sorption and the desorption of the 3.5, because of its larger size, it's not able to get into those pores nearly as well. So when we go in to desorb and knock the 3.5 off, we find that we get most of it to be removed because it's not fitting into those pores. As far as my future studies, I plan to expand this to include both generation 5.5 and generation 7.5, as well as varying the pH levels to see if that makes any sort of difference. I ultimately plan to expand this to become my senior honors thesis. Thank you very much. Does anyone have any questions?
Thank you, Brooke. We're ready for the questions. You're welcome to put the questions in the chat or just raise your hand. Well, everyone thinking, let me ask a question if that is possible. So you kept the pH at 5. However, if you vary pH, you'll get the protonation and deprotonation of the COH group. Would you expect that affect the absorption? And also, so basically, would you expect the charge of the group or to affect the absorption, and how?
Well, so because the ferrihydrite surface is positively charged, if less of those carboxylic acid groups are deprotonated, you would imagine that, so the deprotonation makes them have that negative charge. So when they're negatively charged, more of them are going to be attracted to that positively charged surface. So I imagine when less of them are deprotonated and more of them still have that hydrogen attached to that carboxylic acid group, I would expect that less of it would be able to absorb onto that surface.
Okay, and Jean-Luc is asking a question. I don't know if we can unmute. Let me try to find Jean-Luc real quick.
Yes, Brooke, can you hear me?
Yes.
Nice presentation. I was just wondering, you buy the dendrimers, and if so, how do you know if they are homogeneous?
Yes, so I bought them from Sigma Aldrich, I believe. And they actually came in a 20 weight percent, I believe, for the 3.5 and a 10 weight percent in the 1.5. So, yeah.
Wonderful. Okay. Well, any other questions? If not, let's thank the speaker. Thank you so much, Brooke. And we have now our next speaker in line.
Thank you.
Riley?
I'll go ahead and share my screen.
Mhm.
Okay.
Wonderful. We can see you. Thank you so much. So now we have Riley Burmeister with the talk on behaviors and attitudes of city composting pilot program participants. Go ahead.
Once again, my name is Riley Burmeister. My research partners are Sierra Rice and Kendall Johnson. Our research advisor is Dr. Gina Hill, and we are part of TCU's composting pilot program. Sorry about that. And just to give a little background on our research project is on the behaviors and attitudes of the Fort Worth City's composting pilot program participants. In this program, participants were able to pay a one-time fee of $20. It started in April of 2019, and they were able to receive a 5-gallon bucket for their food waste, and they were able to drop off this food waste at any location in the Fort Worth DFW area to get rid of it for composting. And here we have our purpose and objectives of our study. Our main goal was to describe the factors that were influencing these participants in this composting pilot program and the impact that the food waste was having on their individual homes. And sorry, I think it's on timer. And then on this slide, just to give some background on food waste, 40% of food waste 40% of food is wasted for human consumption, and about 44% of this food waste is from the households. A lot of this food that goes to waste is mainly due on expiration dates, which we'll touch on later in our project, in our research. And these expiration dates are mainly for food quality, and they are not for the spoilage of food. So that's where a lot of consumers are unaware of these dates. And then for the city of Fort Worth, they passed a 20-year resource conservation plan where they aim to divert 60% of the discards from landfills by 2037, and a longer goal of diverting 80% by 2045. And then Sierra.
Can you please unmute Sierra?
I don't see a name, Sierra.
I did.
Okay, sorry about that. Okay. So with the methods, the city of Fort Worth assisted us with the survey development to help create questions for our participants. So these questions included information like their age, gender, race, diet preference, participation convenience, and their motivating factors in taking part in the composting program. The TCU IRB approved of this study, and our data was collected in August 2020, and it was coded and also analyzed using IBM SPSS version 25. So looking at our results, we see that out of 1,054 program participants, that there were 418 total respondents, which approximately achieved a 40% response rate. So on average, the mean age of our participants was around 45 to 46. And within these graphs, the program participants' demographics and the typical diet they consumed were shown. So on this next slide, with this question, the program participants could choose multiple answers for why they wanted to take part in the composting program. The most common motivators were wanting to protect the earth, wanting food waste to become compost, to produce less trash, feeling bad about throwing away food, and being concerned about the city's landfill. Some of the less common responses were being a good example, having peers around them compost, and saving money. So as Riley previously mentioned, the expiration dates are about food quality and not safety. Most food products are not required to have a label by the federal government, so the expiration, sell by, or use by dates aren't a good indicator on food safety. So within the last results slide on the top left graph, we see that approximately 50% of the participants filled their 5-gallon composting buckets within 1 to 2 weeks. So that shows us that the food waste rate is still high. On the bottom right graph, we see that most of the program participants that compost mostly use plastic containers, glass containers, water bottles, and cloth towels/napkins.
Riley, is it you?
There we go.
Who's next?
It's me, yes, thank you.
Okay.
So as Sierra stated, the leading composting motivators were the desire to reduce global footprints and wanting to save the earth, closely followed by guilt about throwing away food and concern over the city's rapidly filling landfill. With almost half the study participants throwing away food based on dating systems, it's no wonder that food waste was high among participants and that nationwide 40% of our food supply ends up in landfills. The only product required to have an expiration date, as they previously stated, is infant formula, and all other dates are only reflective of food quality, not safety. Community outreach and education on food waste reduction methods, date labeling, and ways to use leftovers and shopping behaviors would prove very beneficial as the composting program grows. Fort Worth has a long-term goal of offering curbside pickup of food waste. The program has been a great success. In the program's 22 months of existence, 102 tons of food waste have been diverted from Fort Worth's landfills. And here,
And we have to stop at this point and ask our presenters for the questions. Well, while everyone is thinking of questions, let me ask you all one question about how you mentioned that there is an issue with the food having the expiration dates not when it's spoiled, but when it's actually just loses the taste or quality. What would you suggest? How would you suggest to address that issue so that people don't throw it away and think that it's already spoiled?
I think the main way is consumers have to be educated on the topic. So either it's grocery stores or it's posters, or you could use advertisements to educate the consumers on the actual dating system. And this could be through the grocery store to tell them what those dates mean, or even you could have a side on the label saying what this date used by, sell by means to the consumer and what it means to the grocery store, because they're two different meanings.
Wonderful. Thank you so much. Yes, that's a great idea, and we should definitely pursue that. Let's thank the speakers. And our next speaker should prepare and load, Bong-Han Lee.
Yes, are you able to see my screen?
Yes.
Okay, thank you.
Okay, I would like to introduce Bong Lee with his talk on manganese nitrogen, gallium nitrogen co-doped graphene quantum dots as bimodal magnetic resonance and fluorescence imaging nanoprobes. Go ahead.
Yes. Once again, I'm Bong Lee, and the title was long, so maybe if I repeat it, you'll grasp it. So today I'll be presenting about manganese nitrogen and gadolinium nitrogen co-doped graphene quantum dots as MR and fluorescence imaging nanoprobes. So at the right, you'll see an image, you'll see a cross-sectional image of the brain taken from an MRI. And even for the trained eyes, it can be hard to find for anomalies. So doctors administer gadolinium contrast agents by injection, and to our surprise, we find a tumor in the brain. Although gadolinium ions improve the signal intensity, side effects occur where they deposit in tissues and organs, resulting in diseases such as NSF, where rashes and thickening of the skin occur. To prevent such deposition, gadolinium ions have been chelated in the linear and macrocyclic form. However, leakage of gadolinium ions are still observed. Thus, an alternative form of delivery platform is needed. So in our lab, we have developed biocompatible graphene quantum dots, also known as NGQDs, and they're doped with MRI contrast agents such as manganese or gadolinium ions. Here, NGQDs can act as protective chelators while retaining or even enhancing MRI contrast agent capabilities. And because of the multifunctionality of NGQDs, fluorescence diagnostics are also possible in vitro, which will allow for the tracking of drugs and gene therapies and for some applications in vivo. So for the synthesis, we have utilized a bottom-up approach using a one-step microwave synthesis where we built upon our previous method of synthesizing NGQDs from a glucosamine precursor in water and doped the material with manganese and gadolinium ions. Lastly, we purified and filtrated the final product. So based on our TEM measurements, we have determined their size distributions, which are great values for cellular internalization. And we perform high resolution TEM and find distinct lattice fringes where their lattice spacing is that of graphene. Moreover, EDX was performed showing the atomic percentages of manganese and gadolinium being low. This translates to high biocompatibility. So based on our fluorescence, our nanomaterials exhibit fluorescence not only in the visible, but also in the near infrared, where minor variation of changes are observed. This is nice as it translates to be good for imaging in vitro as well for in vivo studies. So prior to fluorescence imaging, we have determined the highest non-toxic concentration for healthy cells. And with confocal imaging, both nanomaterials have shown to internalize inside cells as shown in green. Three-dimensional voxel image was also rendered to show such internalization. That being said, this added fluorescence capability allows for the alternative without the use of complex MRI techniques. So to gauge for the quality of this MRI nanoprobes, we measured their relativities for their use as positive or negative contrast agents, where positive are the bright regions of the scan and negative the dark regions. And we found that gadolinium doped ones are prone to exhibit more of a T1 positive contrast, and conversely, manganese doped one exhibit dual T1 positive or T2 negative contrast. These values are comparable and even higher than commercial MRI contrast agents. So using a microwave-assisted reaction, we have developed a scalable, water-soluble, and highly biocompatible nanomaterials. And this is important as it solves a critical toxicity issue for gadolinium-based contrast agents, allowing for safer imaging at higher doses. Moreover, imaging is not only limited to MRI, but also fluorescence in the visible and near infrared. And with the multifunctionality of NGQDs, we can deliver drugs and gene therapies in a targeted way, where we can track them during diagnosis and surgery with fluorescence and MRI. So I would like to thank the organizers of this talk as well as my collaborators. Do you have any questions?
Great, thank you, Bon. Let's get some questions. Please post them in chat or just say I have a question in chat or raise your hand. While everyone is thinking about questions, I will ask one. What imaging of what organs do you envision with this?
So in terms of organs, we can look at for graphene quantum dots so far, we can look at intestine, we can look at the spleen, we can look at the kidneys. So with this near infrared imaging capabilities, we are able to image those organs.
Thank you. Any other questions? Okay. If not, let's thank our speaker. And we're preparing now for a new speaker. Olivia, you're here. Okay, great. Great, great. Let's share it a little bit. Olivia, can you share the screen?
Yes. Sharing screen. Okay, you can see that.
Yes, we can see that. Thank you so much. Okay, so it's my pleasure to present Olivia Buchanan with the talk on family functioning and parent mental health in families of color with children with autism spectrum disorder during the COVID-19 pandemic and racial protests. Go ahead.
Yes, thank you. Like you said, my name is Olivia Buchanan, and I chose to study family functioning and parent mental health in families of color with children with autism spectrum disorder during the COVID-19 pandemic and racial protests. I studied this under Dr. Naomi Ikas in the FACES Research Lab. So to begin, autism spectrum disorder is a developmental disorder that's characterized by a broad range of behaviors, including challenges in social interactions, impaired speech and nonverbal communication, and repetitive and restrictive patterns of behaviors. So due to the broad spectrum of deficits and challenging behaviors associated with ASD, parents of children with ASD face many difficulties in raising these children. Studies have found that parents of children with ASD report greater levels of depression, anxiety, parenting stress, and lower quality of life compared to parents of neurotypical children. Although ample research has studied families with children with autism, there's a dearth of prior research looking at family functioning and parent mental health in families of color with children with autism. The current COVID-19 pandemic may exacerbate the stress in families with children with autism due to the disrupted routines and increased anxiety, which for some individuals with ASD can result in increase in challenging behaviors. In addition to the COVID-19 pandemic, the US has also been impacted by national protests against police killings of people of color. The stress of the pandemic, in addition to the current social and political climate, may exacerbate stress in families, particularly families of color. So due to the stress of raising a child with autism being exacerbated by the pandemic and the racial inequality protests, I hypothesized that families of color with children with autism would experience higher rates of stress and have poorer family functioning compared to white families. Additionally, given the lack of research, I wanted to explore concerns regarding police encounters among families of color, as well as their views and experiences of the social justice protests that occurred during 2020. So for our methods, participants for our study were part of a larger study that began before the COVID-19 pandemic. Mothers were contacted shortly after local restrictions were enacted in April 2020 to participate in the study, and they completed surveys in July as well. The majority of our mothers were white, had at least a college degree, they were employed and had a household income of above $60,000 a year. Our mothers of ethnic minority backgrounds were predominantly Hispanic, followed by Asian, and then African Americans. So for our measures, participants completed online self-reported measures of anxiety and perceived stress using the patient-reported outcomes measurement information system. And we measured depression using the 10 dysphoria items from the inventory of depression and anxiety symptoms. And to measure family cohesion during the pandemic, we used the COVID-19 Household Environmental Scale, which provides a tool for measuring essential dimensions of family functioning, conflict, and cohesion during the COVID-19 pandemic. Additionally, to measure parents' marital relationships, parents were asked about the frequency of getting into minor arguments and major arguments. So the results of our study suggest that mothers of color with children with autism had lower relationship satisfaction in April and June, which you can see from the graphs. And this may be because they're not receiving adequate support from their partners. This is also evident in the results that show that mothers in minority families reported higher frequency of major conflicts in their relationships. One reason for this could be, for the lower relationship satisfaction in minority families could be traditional machismo in Mexican American culture, which has been considered detrimental to marital satisfaction. Traditional machismo in Mexican American culture could play a factor in less involvement and support from the fathers due to traditional gender roles. Further research should be conducted to gain more insight into why there's a frequency of major conflicts in minority parents of children with autism. Inconsistent with my hypothesis, minority mothers of children with autism reported lower perceived stress compared to white mothers. These results could be due to extended family support in families of color.
And the findings have implications for the development of services directed to families of children with autism, particularly during the COVID-19 pandemic. For example, focusing on parent mental health and assisting with parenting responsibilities like remote respite care. This may benefit parents and increase family functioning. So the only significant difference we found concerning police encounters was that mothers in minority families were more concerned about their child with autism interacting with police in the future. These findings indicate that because men of color are targeted at higher rates by the police, they're more at risk for having negative interactions with the police as these children of color get older. So thank you so much for listening, and please let me know if you have any questions.
All right, thank you so much. Questions, please. Again, please indicate that you have a question in the chat or just raise a hand.
Kristen, go ahead. I unmuted you.
Okay, I was wondering, could you explain again about minority mothers and the results that you gathered on autism, like severity and functioning?
So the results that we found was that mothers in minority families had lower perceived stress than mothers in non-minority families, which was inconsistent with my hypothesis because I assumed that mothers in minority families would have higher stress, considering that the racial protests, things like that, might influence the higher rates of stress, but this was not consistent with our findings.
Well, other questions are being brought up. I've got a question. Would you think that also the employment and the amount of time mothers spend at work it would influence that? And how would you put that into your study if that does influence?
The question is, does mothers' employment influence their level?
Yes, and basically how many hours a day they work and
right, I'm sure it does. And like you see from our participants, these were like higher SES mothers. So I think it would be interesting to see like lower SES mothers who probably can't afford, daycare or, child services and things like that. And I'm sure their rates of stress would be much higher than these here. So yeah, I would assume that employment rates would definitely influence, stress in these mothers.
Makes sense. Thank you so much.
Thank you.
Okay, if there are no other questions, let's thank our speaker again. And we are moving to our next speaker, Jan Rolf. Okay. Now we have Jan Rolf with the talk on shoreline spiders as sentinels of mercury contamination of the Trinity River. Please go ahead.
Thank you so much for the introduction, and thank you to everyone for being here. My project focuses on methylmercury contamination in the environment, which is one of the most important environmental issues in our world today. Methylmercury is hazardous to vertebrates because it is a neurotoxin in the environment that reduces reproductive success in vertebrates such as humans and birds. Over here on the left, you can see that mercury is emitted into the atmosphere through anthropogenic sources, where it sits in an atmospheric pool of non-toxic inorganic mercury. This atmospheric mercury is then deposited into the aquatic environment, where it is converted by bacteria into toxic methylmercury, which is also the bioavailable form of mercury that can enter food chains. Methylmercury then enters algae at the base of the food web and bioaccumulates as it travels up the food chain. The methylmercury eventually reaches fish, which can pass the contamination to humans, as well as insects, which are eaten by other terrestrial predators such as birds and spiders. Because of widespread atmospheric deposition, all aquatic ecosystems on Earth are contaminated with methylmercury. The millions of river miles of aquatic systems across the globe creates a huge challenge in monitoring the varying levels of this bioavailable contaminant. To solve this problem, scientists utilize sentinel species, which serve to map the bioavailable fraction of pollution in an ecosystem by retaining the pollutants in their tissues. Simply put, they serve as representatives for the amount of pollution in an ecosystem. If we take a look back at this figure that I showed you earlier, you can see on the right side that shoreline spiders feed on emergent insects which are contaminated with methylmercury. And for this reason, they have been proposed as sentinels for mercury contamination of aquatic ecosystems. Shoreline spiders have several traits that make them ideal sentinels. That they are contaminated with aquatic pollutants due to their diet of emergent insects. They are widely distributed, highly abundant, and easily sampled. Most studies thus far have focused on long-jawed orb weavers, while over 30 studies have measured levels of environmental contaminants in long-jawed orb weavers. No studies have used long-jawed orb weavers as sentinels to explore levels of contamination in unmonitored environments. So the objective of this study is to test the hypothesis that long-jawed orb weavers can be used as sentinels to evaluate methylmercury contamination of river food webs. Our study was conducted on 2 forks of the Trinity River, the West Fork and the Clear Fork here in Fort Worth. We picked these sites because a previous study showed mercury contamination in both forks and suggested that the Clear Fork may be more contaminated than the West Fork, which was previously unknown, but they did not have a large enough sample size to confirm this relationship. This raised the question, do the 2 forks of the Trinity River have different levels of mercury contamination? So our approach was to conduct a more robust study of mercury in spiders and fish in the Clear Fork and the West Fork of the Trinity River to see if this relationship would hold true. To do this, we collected 1,150 spiders by hand from both forks of the Trinity River between June and August of 2019. Those spiders were preserved in 95% ethanol. Their leg lengths were measured as a proxy for body size, and the spiders were then sorted by leg length into size categories. They were dried and composited and analyzed for mercury in the direct mercury analyzer here at TCU. Their percentage of methylmercury was then derived from the literature. This figure shows our results. It shows mercury concentration plotted as a function of spider size for the Clear Fork in blue circles and for the West Fork in pink squares. With 5 times the number of spiders from the previous study, our data confirmed that there is a difference in mercury concentration between the Clear Fork and the West Fork, as well as that mercury concentration of the Clear Fork was higher than the West Fork. This suggests that the food web of the Clear Fork was higher in concentrations of mercury than the food web of the West Fork. This hypothesis was tested in a parallel study done by Simon Gall. Simon conducted a study of bluegills between June and August of 2019 and collected between 60 to 75 bluegill in each of the forks. These fish were euthanized in a bucket of MS-222. Their total length was measured. They were sorted into different size categories by total length, dried and composited, and again, their mercury was analyzed using the direct mercury analyzer at TCU. Simon's results are shown here, with the change in mercury concentration plotted as a function of bluegill length. As would be predicted by the spider data, concentrations of mercury in bluegill increased as a function of length in both forks, with a much smaller increase in the West Fork, and that the concentrations of mercury were higher in the Clear Fork than the West Fork. So from this, we can conclude that mercury concentrations in long-jawed orb weavers accurately predicted that methylmercury contamination was higher in the Clear Fork than the West Fork, as confirmed by the study of bluegills. But more importantly, this study is one of the first studies to show that shoreline spiders can be used as sentinels of mercury contamination of river systems. As you can see on the left, long-jawed orb weavers are distributed throughout North America. Therefore, long-jawed orb weavers could be used as a sentinel for methylmercury contamination of aquatic ecosystems, not just in Fort Worth, but all throughout North America. I'd like to say thank you to our honors committee members, Dr. David Minter and Dr. Eric Simonik, as well as Garrett Wallace, Amal Khan, Paxton Churro, and TCU CERC funding. Is there any questions?
Wonderful. Thank you so much, Ian. Thank you for a good talk. Now, questions. Who has questions, please? Okay, we've got several questions. Okay, Meredith, let me unmute you just in a second. Need to scroll. Derek, if you can. Okay. Meredith, you're unmuted.
All right. So, Ian, what do you hypothesize is the reason for higher contamination in the Clear Fork compared to the West Fork of the Trinity? And once you have this information as well, then what can be done to remove that methylmercury contamination, if anything?
Yeah, of course. So I actually had some slides prepped for this. Our professors and we as well have found papers that show that different water fluctuations can affect the mercury methylation in sediments. And so for Benbrook Lake, which feeds into the Clear Fork, is a flood control reservoir. So it fluctuates a lot over the year, which leads to that higher mercury concentration, or we believe it leads to the higher mercury concentration. And Lake Worth, which feeds into the West Fork, is a flow-through reservoir, and it has low between year fluctuations. And so we think that that's why we don't see the amount of mercury, or we see the difference in mercury concentrations. And as for how to remove it, I don't have a good answer for you, unfortunately.
Okay, thank you so much. I think Derek had a question.
My question was, you showed the scatter plot of the mercury growth. My question was, why did you use exponentials, right? My question as a mathematician, why exponentials as your model?
I wish I could answer this one for you better. I apologize for that, but these are just the tests that are typically run in our field. So that's what we chose.
That's a fine answer. Thank you.
Okay, thank you so much. And thank you for a wonderful talk. And
thank you, everyone.
Next speaker should be getting ready to come in. Mitchell Richard.
All right, can you all see this? I'm just gonna hide this. Okay.
Thank you so much. We can see it. Okay, I'm happy to introduce Mitchell Richard with the talk on fluorescent bivalent antibody mimics against epidermal growth factor receptor, EGFR.
All right, well, thank you so much, Doctor. I couldn't have introduced myself better. As he said, I'm actually studying the fluorescent bivalent antibody mimics. So this is something that we have synthesized in the lab. And EGFR is a receptor that is commonly found in some types of cancer, specifically prostate cancer. And it's a pretty big marker for the disease. So it's overexpressed, so it's in a high abundance. And so what we have done is we've taken a synthetic gene for an antibody, so the anti-EGFR, and we call this a Z domain. And so what we've done is we've injected this gene into E. coli, and we can actually grow this in the E. coli overnight so that they can produce this protein in mass amounts. And this is actually a really pretty cool thing, because the current method for producing antibodies is fairly intensive. It's pretty expensive, and it also uses animals. So, you know, as an animal activist would argue, you know, you don't want to inject an animal with a virus if there's a better way to do it. And this is something that we're trying to do, is we're trying to find a better way to create antibodies. And so what we do after this point is we attach a fluorescent molecule to it. And we want to do this because we want to be able to use it into diagnostic purposes. So if we can attach a protein to a fluorescent molecule and then actually track it, if it binds to our receptor, the EGFR receptor, we'll be able to see that it has bound to it and that it is present. And if it's in a high abundance, then it might be a sign that there is cancer. And so to start off with, we had to create the actual dansyl fluorophore. So on the bottom left, you'll see that there's this group called the dansyl chloride. And then we take a linker group, so that's piperazine. And we combine those two and we get a pretty good yield of about 95%. And we get this fluorescent fluorescent linker molecule. So then we can actually combine that with this triazine molecule. And the triazine is very reactive. It's very good for attaching the Z domain to the fluorescent molecule. And so we can combine that to create the fluorescent molecule with the triazine attached. And that's a crude yield of 102% right now, 103%. And that's just probably from impurities, so like the solvent used or water or something like that. And so we can actually confirm the fact that we do have this organic molecule with a proton and carbon NMR, as you can see kind of in the middle right. And again, I'm not an expert on NMR, but we can show that there are aromatic regions within both of these graphs, as well as the methyl groups on the amines. And we can just see the characteristics that would be expected from this organic molecule. And so once we have this organic molecule, we're actually able to take the Z domain from the cells, isolate it from the cells, and we can attach it to the Z domain. So currently, we are at the first step, which we have one Z domain attached to this molecule. And so for the SCase page, on the top right, you can see that we actually have a fluorescent band within the gel. which does confirm the fact that we are seeing something around the relative weight that we would expect to see. But as we go forward with this project, we would actually like to attach a second Z domain to this. And so part of the tricky thing is that we can do this first protein at room temperature, but now we are trying to play around with different conditions that would include pH or maybe a different linker molecule that would make it a little bit easier to add it and actually make it an actual mimic of an antibody so that we can have a high specificity. And so, as I said before, we do know that we have this fluorescent protein, but the mass spectroscopy does confirm the fact that without the fluorophore, the protein weighs about 7.7 kilodaltons or grams per mole if you want to be technical. And whenever we have our fluorescent molecule attached to it, we can see that that very high second peak on the right. And that does confirm the fact that this is something that we are looking for. And that is something that we're going to continue to go forward with. And as I said, this is going to be something that is highly useful, especially to like diagnostic tests that we could use in the future. So with the COVID tests, you know, the antibodies that are the rapid tests, if we can find a way to synthesize something that is a little bit more accessible and something that we can make at a much faster and a much cheaper rate, then this could be something that could be universally used. So I want to thank Dr. Yu and Andrea Guedes for their guidance in this project of mine, and I should say of ours, and as well as the TCU CERC Foundation. And that is all I have for y'all.
All right, thank you so much. Thank you for the talk. And let's hear some questions. Again, please feel free to raise your hands. Okay, Kristen?
Why did you have to create the fluorescent molecule by attaching it with the trizine? If I'm not mistaken, you talked about attaching it with the trizine. Can you just go over that a little bit?
Yeah, absolutely. So it's actually pretty tricky. So the trizine molecule, it is very reactive, and we want to have a way to kind of control it. And so whenever you look at the actual structure of the fluorescent molecule with the linker and the trizine, it actually does give kind of a antibody shape, which is kind of a Y shape. And so with two proteins on there, it's going to have a higher specificity. So we can actually bind a little bit better and also bind, you know, have a higher specificity, I should say. And so having that linker in the trizine, it's just kind of like a chain, you could say. So it holds everything together. So rather than attach a fluorescent molecule to a protein, we attach the protein to a molecule that has a fluorescent molecule on there. So it just is, it allows us to a little bit easier attach a fluorescent molecule while also at the same time having fluorescence so that we can't see that it was bound.
Any other questions? While everyone's thinking, I've got a question. So how do you envision this to work as a sensing mechanism? Would you think of a device or gel-based or implantable?
Yeah, it's actually, I think it's really exciting to think about how we can actually apply this to the future. So one of the current tests we have right now is called an ELISA, and that's a pretty common biochemistry test. And we have ELISA that are very common throughout common life. You know, pregnancy test is an ELISA. And so if we're able to create a protein or a molecule that maybe is fluorescent or maybe has a colorimetric, so it produces a color reaction, we can actually potentially have something to where we can change this protein to sense different types of receptors. So that's why I mentioned COVID, is because once we have this protein, we have the method to attach a molecule, or we have the structure of the molecule so that we can design it and then quickly add it. And so right now we're focusing on the EGFR, but we can also target like the beta amyloid protein that is found in Alzheimer's. So we can use that as a diagnostic test. So if it would be commercially available worldwide, I can't speak to that. You know, I mean, it really depends on how we have to make it and if it's viable or commercially. But going forward, we can use a lot of different ways. And I would say an ELISA would be an example of how we would use that in a more broad and available term.
Okay, thank you so much. Let's thank our speaker. All right, and our next speaker, Kaylee Decker, should be getting ready.
Yes, let me share my screen real quick. Okay, can we see the screen?
Yes, thank you so much. It is my
Awesome.
pleasure to introduce Kaylee Decker and Charles Lord with the talk on the effects of trait extrapolation on attitudes toward kneeling during the national anthem. Thank you so much.
Yes. All right, thank you for the introduction. Yeah, so my research is over trait extrapolation and how that can influence our attitudes towards different topics. And the topic specific to this presentation is attitudes towards kneeling during the national anthem. So this all sounds great, except for what does extrapolation mean, right? In this context, we look at extrapolation and we think of it as making an inference based on what we already know. And in our paradigm, we look at attitudes as being flexible and adaptive and informed thoughts come to mind in that moment. So if you tie the two together, we have trait extrapolation, and we'll say in general, it's just about a group. Someone may know that a group has certain attributes, such as being friendly and predictable and generous. And then we may extrapolate beyond what we know to information that is self-generated that we're coming up with, and say that they're also considerate and they're charitable and they're genuine. And then in turn, this could influence how we evaluate that group later. Now, our research has looked at extrapolation and trait extrapolation specifically in a number of different ways towards a number of different topics. But the current presentation was interested in whether extrapolating about people who agreed and disagreed about a social issue, so such as kneeling during the national anthem, whether that would polarize or make people become self-radicalized towards not the people that they extrapolated about, but about the issue in general. So in other words, can extrapolating about people who agree and disagree with you change how you think about a topic that you agree or disagree about? And for this project, we had 171 participants. We first asked them whether they supported or opposed kneeling during the national anthem, and we also asked them the extent to which they supported or opposed this issue. And then we randomly assigned them to either an extrapolation or a control condition. For the extrapolation condition, we just asked them what 5 traits would come to their mind when they thought about proponents of kneeling during the national anthem. And then we also asked them what 5 traits would come to mind when they thought about opponents of kneeling during the national anthem. Our control condition just typed different synonyms for various words, but we had 5 positive traits and 5 negative traits that they typed synonyms for. And this was used so that both conditions were typing traits and both conditions were specifically typing the same number of positive traits and negative traits. Our dependent variable, the only one I'm really going to discuss in terms of this project for the purpose of time in this presentation is the one here at the bottom. So what is your overall attitude towards kneeling for the national anthem? We also asked questions about people who agreed and disagreed with the participant on their stance, but like I said, for the sake of time, we'll just go over this one right here. So our question was, did extrapolating about proponents and opponents of kneeling cause self-radicalization towards kneeling in general, right? Not towards the proponents and opponents, just towards the topic of kneeling. And we found that people who initially supported kneeling during the national anthem indicated a slightly greater support for kneeling during the national anthem after extrapolating compared to our control group. And we also found that participants who initially opposed kneeling during the national anthem were more likely to oppose kneeling after extrapolating than the control group, and this was highly significant. So what's next for us? Well, we want to determine whether the generalization effects, or whether these effects generalize to other issues, whether they be social issues or political issues and things of that nature. We also want to examine whether there may be important individual differences in who might be most likely to self-radicalize or who would self-radicalize the most when they extrapolate. And then also we would like to explore whether we can see the same effects if people use a different type of thought strategy rather than extrapolation. So extrapolation is just one instance where people can go beyond what they know about a group, right, or a topic, or whatever the attitude object may be. But there are likely, and definitely are, other ways that people do these things, right? So one possible way might be communicating online. When we write a social media post, we're doing something very similar. So we'd like to explore these avenues as well. And I would like to thank you for letting me share some of this research with you today, and I'm open to any questions you may have.
Great, thank you very much for the talk. And let's see some questions. Let's hear some questions.
Julie, I unmuted you if you had a question.
Oh yeah, I was just curious about why you made the distinction between the act as a whole, like kneeling, as opposed to the people who do it.
We were just interested in exploring different social issues. And in terms of this, we had actually conducted another study right before this, looking at the same thing, but with a different issue. And we were trying to think of something that was particularly salient at the time, but we didn't have really a reason for that distinction or that justification.
While everyone is thinking about questions, I've got one. This is an interesting approach, and I'm just thinking of which other social or economic issues could you apply it to? Which ones are on your mind? What's coming next?
Yeah. So right before we conducted this study, we actually looked at attitudes towards people who agree and disagree with the extrapolator about legalizing abortion and things like that. And we didn't actually measure attitude towards abortion. And I was talking to my mentor, Dr. Lord, and I said it would be really interesting if we could see, because we found similar effects where people were becoming self-radicalized after extrapolating towards people who agreed and disagreed with them. Of course, positive for agreeers and then negative for people who disagreed. And I said, well, I wonder if this could also influence how they think about the issue in general. So we'd like to replicate that study. For one, but there are also several other issues that are out there right now. Gun control, right? We could talk about that. We could look at people who agree or disagree with gun control or making guns more strict, things like that. But we try in general to mirror popular issues in society.
Wonderful. Thank you so much, and good luck with your work.
Thank you.
Speaker again. And our next speaker, Dang. Hello.
Yeah, I'm sorry. I'm trying to share my screen. Okay.
All right. Wonderful. So I'm happy to introduce Dang with his talk on wound healing process modeling using partial differential equations and deep learning. Go ahead.
Hi everyone. So my name is He. First, thank you for your great introduction. And I'm a senior majoring in math and computer science at TCU. And I want to talk about research projects that I have opportunity to participate with Dr. Richardson in math department. The project is called modeling wound healing using partial differential equations and deep learning. So in this talk, we will discuss about the motivations, our approaches, and some results. The motivation of this project is really straightforward. I believe everyone here had some scratches and some wound before, and we want to know when the damage is healed and the process of it. And the problem is different patients would have different wound healing processes. For example, patients with a background of diabetes may have difficulties in healing. For this reason, by understanding this process, we can provide and determine the appropriate amount of medicine to give to patients with varying type of wound. So for our approach, we want to discuss about the workflow, some math models, deep learning models used to handle the data set, and one again, the workflow. So there are several concepts that I want to walk through in this project. First, we use a variant of alternating direction implicit, or ADI, which is an approximation method to solve math models of the wound healing process. Second, to handle the wound, we need to have some images of those. However, how can we identify the wound in the image? We use a deep learning model to segment or identify the wound from the pictures. So for a better explanation of the process, this is the entire workflow for the project, such in the simple pictures. First, we have the wound image. Then we have the deep learning model, and then we have the results after the deep learning model in the middle. And in the last pictures over here, after putting the middle pictures into the modified alternating direction implicit, we got the result. And this is the result of the wound through the modified ADI after 14 days. So now let's talk about the math models. We believe that we could use a math models to determine the wound healing process. And in this case, we use diffusion equation to model wound healing. And we have some parameters. First, we have the threshold theta, and we determine if the areas of the wound in the image completely heal or not. Another parameter is K, which represent in the equations over here. We represent the speed of the wound healing process. We determine like if the wound healing is fast or slow. And we also have another parameters which represent resolution of images, delta X, delta Y, and delta T. So we develop an algorithm based on alternating direction implicit, an approximation method to solve these equations. We have chosen ADI because of its stabilities in solving the equations. And this is how ADI compares to other approximation methods, and one of those is called Euler's. And you can see here's ADI outperformed Euler approximation method. So we have talked about how we could model the wound healing process using PDE. We now need some initial condition or input to the PDE of solutions. So as mentioned in the workflow, and we come to workflow again, we need to identify the wound and retrieve some information from the images. So we need the middle pictures over here. This is called segmentation problems. And we believe that deep learning is an excellent way to do that when we could solve this problem by analyzing pictures that we have. And in our case, we implemented UNet because of its effectiveness. And this is an example of the deep learning result that we have. As you could see, the UNet models outperform the residual UNet model in segmenting the wound from the image for this type of data set. After all of these concepts, how could we predict the wound healing process? This is when the parameters I told about come into play. The two pictures at the top are the output from the deep learning model of patient real images. The left one is when the wound at day 0, and the second one is the wound at day 14. And the bottom three pictures are different results with different parameters combinations. And in conclusions, what is our results so far? We have proved the stabilities of alternating direction implicit and modify it to fit our projects. And we have built a deep learning model to segment or identify the wound in the pictures. And our next step is to find suitable parameters K and theta to capture exactly the wound healing process. I want to say thank you for you guys to listening to my talk, and thank you, Dr. Richardson, for advising in these projects. Thank you.
Thank you so much. Let's thank the speaker. And let's see some questions.
Ian, I unmuted you if you had a question. I was wrong. Guess not.
While everyone's thinking, I've got a question. General question about the biomedicine.
Can you say again? I think it's a little bit cut off.
How would you apply this in biomedicine? How would you think of future biomedical applications of your work?
Oh, yes. So the problems identified at the beginning of my talk is different patients. We have different type of wound and like wound healing process, and they many people. So we cannot give different patient like this the same appropriate of medicine for typical the wound. Even they have the same wound, we cannot give the same medicine or something like that. So if we can predict ahead of time, like what is the wound healing process is with this type of medicine, we can give them appropriate amount, and then we can give them different type of wound and then different patients.
Okay, that makes sense. That's great. And maybe we could actually already screen the wound like that and do the some automatic healing approaches with automated medicine in the future. So there's a lot of applications. That's awesome. And we have another question, actually same question from Derek and from Becky. Let me try to unmute Derek or Becky, whoever would like to go first.
I was just pushing Becky's question out, but she said, why did you select the heat diffusion equation?
Oh, yeah, because we believe that the wood healing process is like performed by the diffusion, much like the diffusion equations. So when the wound diffuse in the surface of skin, so and we have researched a lot of topic and papers related to this problem, and they may also use the type of equation. So we want to give it a try.
Sounds good. Thank you so much. Let's thank our speaker and move to our next speaker. Jae-Hyun Lee.
Hi.
Hello.
I will share my screen real quick. Good afternoon, everyone. My name is Jae-Hyun Lee, and today I'll be presenting over my researches over the effects of BRCA1 protein length on DNA repair mechanism. So BRCA1 is known to be a tumor suppressor protein when there is a damaged DNA, whether the DNA damage has been whether the DNA has been damaged due to chemical toxins or UV radiation. BRCA1 is crucial in maintaining genetic integrity and suppressing tumor proteins. In this mechanism, DNA damage repair mechanism, BRCA1 does not stand alone in its function. POLB2 protein has to be recruited together, and they both have to interact in a proper manner in order to be recruited to the DNA damage site and repair the DNA properly. However, the interaction between those two proteins, the binding interface is largely unknown and their structure is largely unknown. So our objective was to investigate the secondary structure of BRCA1 upon binding to POLB2 protein. In their inactive state, POLB2 exists as a homodimer coiled coil state, whereas BRCA1 binding interface to POLB2 remains largely unstructured. So upon DNA damage, there are two options that can come from, two outcomes that can come from it that we suspected is that BRCA1 protein could either remain unstructured upon binding with POLB2, or BRCA1 can take up a coiled coil alpha helix structure upon binding with POLB2. So our project goals consisted of testing whether the length of BRCA1 affect binding between BRCA1 and POLB2 using the nuclear magnetic resonance experiment. And if that were to confirm our hypothesis of BRCA1 protein forming a coiled coil alpha helix upon binding, we wanted to investigate the exact conformation of the protein-protein complex. So this is our results, one of our results for NMR graphs. So if I were to explain this briefly, the y-axes represent the hydrogen of the backbone of the protein. So right here, the hydrogen and nitrogen interaction between the amino acids, and the x-axis represents the nitrogen atom right here. So here we see the BRCA150, which is a truncated version of the protein that we have made from BRCA191 amino acid length. We see that in the black peaks, which represent just the sole presence of the BRCA1 protein in the system, we see that it is heavily contoured and heavily located in the central region of this graph. Whereas when POLB2 is added to the system, which is represented by the red peaks, the spanning of the regions are much broader and larger, and the contours are a little bit lessened. So what contours represent is that when the contours are lessened, this represents then flexibility and rigidity of the overall complex. So we were able to induce that upon introduction of POLB2, due to the decreased contour, we were able to induce that they form a secondary structure, and due to the large spanning of these peaks, which represent the change of the chemical environments of these nitrogen and hydrogen atoms, we were able to induce that our hypothesis of BRCA1 protein forming an alpha helix coiled coil protein was supported. So if I were to give you guys another picture and compare the original non-truncated version of BRCA1 91 amino acid formation, you guys could see that whether POLB2 is present or not, the nitrogen and hydrogen region on the NMR graph is pretty still heavily located in the middle region, and the contour has really not changed that much, meaning that the length of the BRCA1 protein significantly has some sort of an impact of the secondary structure or the BRCA1 POLB2 complex formation. Thank you. I would like to answer any questions if you guys have any, and I would like to thank Dr. Stewart for all her wisdom throughout this research.
Okay, let's thank the speaker. And we're waiting for some questions.
I muted you if that was a question. I'm not sure if that was just a round of applause.
Oh no, that was just applause.
Well, they're warranted then.
Okay, well, everyone is thinking of questions. I guess my question is, what are the further steps? How are you going to verify it in biological environments?
Sorry, could you repeat that again?
Sorry, what are your further steps and how are you going to further work with it and verify it in biological environments?
We could perform an ITC experiment. I believe that is not something that I have investigated or learned yet, but that is another method that we use in the lab that can confirm the further interaction. Or we could use other, I believe, that's something that I could think on top of my head, that we can look at another protein interaction or any energy, I guess, related, like the heat release and so the interaction binding between proteins.
Yeah, that could be a very interesting one. Let's see, any other questions? Okay, if not, let's thank our speaker again, and let's thank all of our speakers in this session. This concludes the current session. Thank you so much for being here and participating and contributing wonderful, super interesting, exciting talks. Thank you everyone for being here. Bye-bye. See you all at the awards ceremony.